My name is Burks. I'm a senior cadet at Magna High School. Today I'm here to present to you the effect of freshwater acidification on clams. The impact of climate change and the increase in carbon dioxide in the atmosphere is the largest debated topic during the century. The oceans have increased in acidity by more than 30% in the past 200 years, and this has a harmful effect on saltwater organisms. However, not much is known about the effect of increasing carbon dioxide on freshwater organisms, and even more, not much is known of how increased carbon dioxide affects freshwater clams, especially tuberculous clams. When freshwater absorbs carbon dioxide, a chemical reaction occurs that produces corrosive carbonic acid. Carbonic acid separates to make hydrogen ions and also detrimental bicarbonate ions. These additional hydrogen ions will then go on to bind with other carbonate ions to make even more bicarbonate ions. And this chemical process leads to an increase in aqueous carbon dioxide, but a reduction in carbonate ions. If more and more carbonate ions are being consumed by the bicarbonate and hydrogen ions, then there's less carbonate to make calcium carbonate, which is a very vital nutrient and resource that shell-building organisms, such as clams, require to build and grow their shells. I hypothesize that there's an increase in carbon dioxide in the water of freshwater clams, then they will impede their growth because they do not have the appropriate nutrients to grow their shells. Previous studies done on saltwater acidification or ocean acidification showed that the effects of saltwater acidification on organisms are very varied. For example, in one study, it was found that with shell-building organisms such as lobsters, clams, and mollusks, and also crabs, that additional carbon dioxide in their water actually impeded their growth and caused a decrease in their mass. However, in some algae species and saltwater environments, an increase in carbon dioxide correlated to an increased population growth. Other laboratory studies done on particularly freshwater acidification have shown that freshwater acidification is actually acidifying and having a more rapid effect than ocean acidification. For example, one novel study from 2015 over a 35-year period has shown that the pH of the freshwater environments actually decreased 0.3 units. And this is a very small number, but actually the pH is logarithmic, which means that this is actually a very huge number. And in contrast, the pH of the oceans is decreasing by only 0.1 units in the last 200 years, which further shows that the freshwater is acidifying at a very alarming rate and a lot faster compared to saltwater environments, which then shows that this high freshwater acidification needs to be further studied. When it comes to the effect of freshwater acidification, there's not been many studies done on how it affects organisms. There's one paper done on how it affects the defense mechanism Daphnia, and showed that the defense mechanism was actually lowered when there was increased carbon dioxide in the water. On the other hand, one study done on freshwater algae showed that there was an increase in the population, and also found that freshwater fish and also other amphibians, that they had a decrease in growth and population size. In order to conduct my experiment, I first obtained 4 15-gallon freshwater aquariums for the clams designed during the experiment, with one taken as a control. I then calibrated each instrument, and I built a custom apparatus, which you can see in photo 4, which was connected to Arduino microcontroller and also utilized solenoids and Aquamanager software. This apparatus was also connected to carbon dioxide tank, which we use to distribute carbon dioxide among the tanks using the carbon dioxide bubbler shown in photo 3. I used carbon dioxide in order to decrease the pH and make the water more acidic, while also connected the apparatus to a flask baking soda, a sodium bicarbonate, in order to make the water more basic. I then changed the pH of my tanks to the ranges of 6.5 in tank A, the acidic tank, 7.5 in tank B, the neutral tank, and 8.5 in tank C, which was the basic tank. I used these numbers in order to simulate the decline of freshwater pH in 200 years, which is decreasing, as said earlier, by 0.3 units. I then measured the mass of the clams in their individual containers, and I also put them on containers to measure their dissolved oxygen and dissolved carbon dioxide, or the respiration. I then put 22 clams in each of the tanks, and I let them live there for around 3 weeks. And then after that, I recorded their mass, and once again, the dissolved oxygen and carbon dioxide. and I compared all my data using a parametric ANOVA test in order to see differences in mass, and I also conducted a Kuska-Wallace test to see if there were changes by separation. There's also a study where very conclusive will be found in ocean acidification. In graph 1, it shows the before and after change in masses of the clams. The clams with pH of 6.5, the acidic environment, displayed a % decrease in mass, and so did the clams in the neutral environment with a pH similar to 0 to 7.5. However, this change in pH was less drastic. However, in pH of 8.5 in graph 1, clams had a slight increase in mass, which I found very interesting. And this data right here shows that as the water became more acidic, the clams started to lose mass, which correlates to the data found also in ocean acidification. In graph 4, it shows the average % change mass of the clams again, where the clams in the acidic environment had a drastic decrease in mass, while the clams in the basic environment had a slight increase in mass in the pH of 8.5. In graph 2, it shows the average change of the level of oxygen of the clams in the environments. It's very evident that the clams in the acidic environment have far less oxygen consumed compared to the clams in the basic environment, while clams in the neutral environment with a pH of 7.5, they had a little bit less oxygen consumed than those of their basic counterparts. This shows that clams in the acidic environment, they're consuming a lot more oxygen than clams in the basic environment, which is an indicator of stress and also an exertion of energy to build their shells called freshwater acidification, which I said earlier caused a decrease of the clams in the acidic environment to be able to obtain calcium carbonate to build their shells and to grow. So to make it more simple, the clams in the acidic environment, they're basically running a marathon and gasping for air, while clams in the basic environment, they were doing a light jog and pretty comfortable in the environment. In graph 3, it shows the average change in dissolved carbon dioxide by pH among the clams. And dissolved carbon dioxide is merely how much carbon dioxide was being respired by the clams in the environments. The data showed the clams' the acidic pH of 6.5, they had a significant increase in carbon dioxide compared to clams in the basic environment of 8.5, while clams in the neutral environment had less carbon dioxide than clams in the acidic environment. When these dissolved oxygen and carbon dioxide are well compared, this shows that the clams in the acidic environment, they were under a lot more stress than those in the basic environment. And even the neutral tanks, 7.5, as you can see in dissolved carbon dioxide levels, they had the effects of kind of an average between the acidic environment and basic environment. The clams in the neutral environment, they did lose some mass, but they also had more carbon dioxide in their water than those in the basic environment. So this shows the clams in the basic environment, they're pretty much stable in their pH due to it not having a lot of carbon dioxide. While clams in the acidic environment, they're really stressed in their environment and not happy. The percent decrease in the mass, as I said earlier, was due to the fact that not enough calcium carbonate ions in the water. And carbon dioxide, while clams in the acidic environment were not able to build and maintain their shells due to an increase in carbon dioxide, which resulted in a decrease in calcium carbonate. And even though this experiment was conducted for a pretty short amount of time, drastic results were seen. In the future, I'd like to conduct the experiment for a lot longer, maybe over the course of a couple of years, to see if clams actually adapt to the environment, which could therefore change the results of this experiment. There have been multiple studies done on how ocean acidification affects oysters, clams, scallops, corals, starfish, and many other organisms. However, according to my research, only around 1 to 3 studies are done on freshwater acidification in respect to shrimp and also daphnia, and also amphibians and fish. And even with these studies, the results were very varied, with some organisms having increases in growth, with others having decreasing growth. And this uncertainty with how freshwater acidification can affect organisms led me to conduct this project. I hope to use my data in the scientific community to show that a pH of 7 or 0 or less can actually have a negative effect on clams. And with ocean acidification already having a negative effect on many organisms, I hope this research can help spur more research being done on how freshwater acidification can impact other organisms, such as corals and other kinds of clams and lobsters. Freshwater composes around 3% of the water on Earth, and the rest being saltwater. And even though this is a very small percentage, this does not mean that freshwater should be overlooked when it comes to research. Regardless of freshwater being less than 5% of the water on Earth, it is still what more than 100,000 species of freshwater organisms call home. There are over 800 freshwater mussels and clams that reside in America, but I predict that in around 20 years from now, as the pH units in the freshwater decrease to below 0.3 units, these numbers of freshwater organisms and shell-living organisms will be drastically reduced, and they'll probably be endangered as freshwater acidification becomes more apparent, and also as the amount of carbon dioxide in the atmosphere continues to increase, and it dissolves our freshwater. I believe the results of this study can greatly benefit national environmental agencies and also local agencies, and potentially help raise more awareness about the growing problem of freshwater acidification. If more awareness is done on this topic, then this can lead to greater action to reverse freshwater acidification and reduce our carbon dioxide output, which is a very viable solution to this problem. It's been more than 200 years since the amount of carbon dioxide atmosphere is rising, and its rate of the amount of carbon dioxide in the atmosphere now is at the highest point in past years. And this can greatly affect freshwater organisms such as clams. If there's a decrease in clam population, this will also affect other organisms feed on clams, such as sea stars, fish, shrimp, and crabs, and even humans. The freshwater seafood industry is worth more than $340 million, but the revenue will soon decline if clams and other shellfish do not have the appropriate nutrients to grow their shells due to increased carbon dioxide levels and also anthropogenic greenhouse gas levels. This decline will not only affect the freshwater seafood industry, but also affect farmers who rely on freshwater clams and also fish in order to feed on clams. And this increase in freshwater acidity will also affect fish that families rely on to feed their clams and also to feed their families and make money from themselves, which account for more than 1 million people in Alaska and other Arctic regions. And then millions of people live in rural areas that are near freshwater ponds, streams, and lakes. And freshwater acidification is not addressed, that can potentially harm these people and how the many vital resources that they rely on to live. Therefore, even though freshwater is only 3% of the water on Earth, I would strongly believe that this issue needs to be addressed and studied more in order to help a growing freshwater industry and millions of people who rely on freshwater clams and other organisms. In conclusion, this experiment has shown many things in the area of freshwater acidification. A pH of 7.0 or less has negative effects on the growth of bigothalate clams. It also proved that as the water becomes more acidic, the clams gradually lose their mass and start to increase their respiration rate, shown by an increase in carbon dioxide but a decrease in oxygen in the clams of the acidic tank. The apparatus that I constructed was also deemed successful in this area study and can also be used in a variety of other studies related to marine organisms. And it is now known that increasing carbon dioxide has a negative effect on the growth of bigothalate clams. And I believe as global anthropogenic greenhouse gases increase, this poses a threat to clams and other organisms. And these findings of the study continuously change how much research you put in freshwater acidification, because right now we are very far behind. There could very well be one day where somebody walks into their local restaurant and ask for clams or oyster or any other kind of freshwater or even saltwater organism, but they can't get any because they're all gone. And this is one of the reasons why I think this project is important and why freshwater acidification deserves to be further studied. I'd like to thank Louisiana State University of Shreveport, Dr. Amy Erickson, and also Mr. Christopher Stratton for helping me obtain my resources to conduct this very successful experiment. That is my presentation, and I hope you enjoyed listening. Thank you.
Awesome. Thank you so much, Elijah. Let's check Amy, Greg, if you guys want to hop on video. Greg, if you're here, I know you had a question prior. If you want to go ahead and hop on and ask now.
Yes, I'm back. Thank you, Elijah. I'll just repeat what I started to ask before, and that was that I've seen the research that you're talking about on the effects of acidification on aquatic organisms in freshwater systems, but I think there's an important difference between the way the previous research was done and the way that you're doing it. You're addressing the question of acidification due to increased concentrations of carbon dioxide in the atmosphere. And so, and I like the fact that you're doing this realistically by, are you bubbling CO2 into the aquaria or something to get this done?
Yes.
Which is a great way to mimic that environmental concern. Acidification. I've always heard it called ocean acidification because most of the research has been done in the oceans, but of course it's going to be affecting freshwater systems as well. Well, probably 30, 40 years ago, people were very concerned with this phenomenon called acid rain, which also lowered the pH of freshwater systems, but via a very different mechanism. You're directly adding fairly strong acids into the water, and that's the way those experiments were done. And so those are different methodological approaches which affect pH, but affect the carbon cycle differently. And I haven't had an idea to really think about the consequences of these methodological differences, but I was just wondering if you've had more time to contemplate that, and maybe you can speak to either the applicability or lack of applicability of previous studies on acid rain deposition versus acidification due to atmospheric carbon dioxide. That was a long-winded question, so I will for now.
Well, from what I've read in different studies, one study I read that the acid rain kind of project, the problem with that project is that acid rain can be from different things, such as like leaching and stuff into the waters. And this was like kind of one pitfall of my project was too, is that I kind of more manually added the carbon dioxide into the water, which doesn't really mimic how it will happen into the environment realistically. But I try to like add the carbon dioxide slowly over a long period of time, and like not just add it all at one time. It's like not shock the clams.
Thank you. Yeah, that makes a lot of sense.
So Elijah, I know that you won some awards. Elijah is a high school student, actually. Yes, and so can you tell us about the competition that you were involved in, and then the additional research that you have pursued? Because the research we did, I think, was back in fall of 2019.
Yeah.
So when he's continued to do work. So can you first tell us again about the competition and then about your current research?
Well, the competition my sophomore year of high school for this project, it was very fierce. This was like my first time going into like the science world and like working with the lab for the first time. And so the experience was definitely life-changing. And because of that, I got first place at the school level, reaching those in the state science fair. And also got a bid to international science fair too, because it's one project. And so being able to present the research was a very big part of my high school experience. I won some money too over this research also. And so that was pretty fun. And right now, I did one project last year on the effects of nitrous oxide on crawfish. And that was kind of like a continuation of this research with different gas, see how that would affect crawfish, which is Louisiana kind of staple food. And so basically this one project really helped kind of spearhead my world into spearhead my experience with science world.
Fantastic. That's great. Thank you for sharing that with us, Elijah.
Elijah, congratulations. That is extremely impressive. I assume you were a master's student or an undergrad, but somebody to be doing this quality of research as a high school student, it's young people like you that give me hope about the future. So thank you very much. Thanks for bringing that to our attention, Amy.
Sure.
Thank you so much, Elijah. Yeah, your presentation was so informative and very impressive to know that you are in high school. And wow, I can only imagine what kind of research you'll be doing in the next few years. That's great.
Thank you.
All right. Are there any more questions from the audience? Anybody that wants to hop on video, audio, ask a question, or make a comment to Elijah? Just give it a few seconds. Okay, well, if there are no more questions, we're going to go ahead and move on to the next presentation. Thank you so much, Elijah, again. And I'm going to go ahead and get set up for the next presentation. Thank you. Thank you. Thanks, Greg, for your questions.